Lung adenocarcinoma, or LUAD as we'll call it, is still one of the most lethal cancers we face globally.
It really is. The numbers are staggering.
But there's this paradox, right? It's something that actually gives researchers a lot of hope. LUAD doesn't just appear out of nowhere.
No, it develops over time. It typically emerges from these recognizable precancerous lesions.
Things like atypical adenomatous hyperplasia AAH and adenocarcinoma in situ, or AIS.
Exactly. And that slow progression, in theory, presents this phenomenal window of opportunity for us to step in.
To stop the cancer before it really even starts. But that's been the huge challenge.
It's been a frustratingly blurry window. We can see the precursors, but we don't know which ones are going to progress. We don't know how that cellular transition actually happens.
And without that map, you can't really intercept anything effectively.
Precisely. For interception to work, you need a high-resolution roadmap of the entire journey.
And that's exactly what we're diving into today. We are breaking down a study that created, I think, a definitive, high-fidelity spatial roadmap for how LUAD gets started.
And they didn't just sequence the cells, which is key. They mapped them geographically right inside the actual human tissue.
The technology they use for this sounds incredible.
Oh, the tech stack was immense. We're talking about multimodal spatial-omics. So they're combining data from, I think it was 56 human precursor and invasive lesions.
Wow, from 25 different patients.
A really comprehensive data set. And for listeners, when we say multimodal, it means they use several different powerful tools in combination.
So things like single-cell sequencing, but also spatial transcriptomics.
Exactly. So single-cell RNA sequencing tells you the exact program running inside thousands of individual cells. But then they layer on things like Visium and this ultra-high-resolution tool called Xenium.
Okay, let's unpack that. If single-cell tells you what the cell is, what do Visium and Xenium do? How do they give you the location, location, location?
It's kind of like having a satellite map and a street view camera all in one. Visium overlays gene expression on the tissue, so you see general neighborhoods of activity.
Like a heat map.
A sheet map, yeah. But then the Xenium zooms all the way in. It gives you single-cell resolution within that tissue context.
So you can actually say this specific type of cell with this program is physically touching this immune cell.
That's it. And that level of certainty allowed them to identify these co-evolutionary partnerships. It's not just about what cells are there, but who their neighbors are.
And the most important partnership they found, the core insight really, was what they call the epithelial-proinflammatory niche.
Correct. This is the driver. The whole process starts with specific alveolar progenitors, the epithelial part, that get sort of co-opted and pushed forward by their immediate immune environment, the pro-inflammatory part.
Okay, so let's start at the absolute beginning. If we're looking for that cellular seed of LUA, that very first cell type that goes rogue, where did the map point?
It pointed directly to a cell subset they identified molecularly as KRT8 high alveolar intermediate cells, or KACs for short.
KACs.
And pathologically, these KACs correspond perfectly to something clinicians already recognize under the microscope. Reactive pneumocytes type 2, or RPII.
So KAC is the molecular name, and RPII is what a pathologist would call it.
They're the same critical cell.
So why them? I mean, aren't alveolar type 2 cells, the AT2s, just the normal repair and regeneration cells in the lung?
They are. AT2s are the lung's stem cells.
Yeah.
But these KACs they're an aberrant intermediate state. The single nucleus sequencing placed them transcriptionally right between a normal AT2 cell and a full-blown tumor cell.
So they're halfway there, genetically speaking.
In a sense, yes. They still had some normal AT2 features, like expressing a surfactant protein called SFTPC, but at the same time, they were co-expressing these really clear danger signals.
You mentioned these were metaprograms, MP6 and MP7. What does that actually mean? What software is the cell running?
Right. So think of a metaprogram as a bundle of coordinated software. MP6 is the tumor-associated program.
So cell division, growth.
Exactly. All the machinery for uncontrolled proliferation, for ignoring the normal limits on growth. And then you have MP7, which is the inflammatory or stress program.
Something is wrong here.
Yes, exactly. It's like the cell's internal alarm system sensing damage, initiating stress pathways. So these KACs are basically running the start dividing software right alongside the I am stressed and damaged software.
So they're primed for trouble. But did the researchers confirm that these KACs, these RPIIs, were truly the first step?
They did, using something called spatial clonal analysis. It's like genetic ancestry testing for cells in the tissue.
Cracking shared mutations.
Exactly. And they found that RPII cells often mapped to the very earliest, the initial clones. sometimes even in clones shared between what look like normal tissue and a visible lesion.
That's a huge point. It suggests the problem is already there before you can even see a lump or a nodule.
It really is. It supports this idea of a field effect, where the precancerous change is already seeded throughout the surrounding tissue, just waiting for the right trigger.
Which brings up the next question. If these KACs are everywhere in this field, what determines if they just stay reactive or if they actually turn malignant? What's the trigger?
That's where the neighborhood comes in. The inflammatory niche becomes the crucial determinant. The KACs are almost passive at this point. It's the microenvironment that actively pushes them over the edge.
So what did the spatial mapping show? Who are the neighbors of these KACs in the early lesions?
It was incredibly clear. These precursor spots were just packed with pro-inflammatory immune cells. And one type in particular stood out, IL-1B high macrophages.
So specific inflammatory macrophages.
Very specific. The data showed this perfect positive correlation. Wherever you saw the gene signature for a precursor cell, you saw the gene signature for these myeloid cells right there with it.
Okay, so they're hanging out together. But correlation isn't causation. What's the specific signal? What's the conversation happening between the macrophage and the KC that flips the switch?
The conversation is the IL-1B, IL-1R1 axis. This is the lock and key mechanism that drives the whole thing.
Okay, break that down for us.
Sure. The key is the ligand, a molecule called interleukin-1 beta, or IL-1 beta. It's being pumped out primarily by those macrophages in the niche.
And the lock?
The lock is the receptor, IL-1R1. And here's the kicker. That receptor is uniquely and highly expressed only by the KACs and the precursor cells.
Wait, so normal lung cells don't have it?
Not at all. Normal alveolar type I and type 2 cells completely lack this receptor.
Wow, so that inflammatory signal, that IL-1 beta, is like a guided missile. It only hits the precancerous cells.
It's an incredibly targeted signal. An inflammatory response that should be protective is instead sending a targeted pro-growth signal only to the cells that are primed to become cancerous.
And what's fascinating is the timing of it all. This whole engine, this niche, is really active in the precursor lesions, right? The AAH and AIS.
Highly prevalent, yes. But it becomes much less frequent as the tumor becomes a fully invasive LUA.
What does that tell you?
It strongly implies its main job is initiation and early progression. It's the match that lights the fire. It's not the fuel that keeps the big bonfire going later on.
Once the cancer is established, it finds other ways to grow.
Exactly. It switches to other drivers, making that initial IL-1B, IL-1R1 axis kind of irrelevant in the late stage. And that timing is absolutely crucial for any kind of interception strategy.
Okay, so let's bring in the genetics. How does this whole inflammatory partnership play with the big driver mutations we know about in LUAD, like KACs?
The link with KACs is extremely strong. I mean, it was one of the clearest findings.
What did they see?
When they sorted the data by KACs mutation status, they found that in the KACs mutant cases, that IL-1B, IL-1R1 interaction was markedly more prevalent. It was present in 100% of the precursor lesions they analyzed.
100%.
100%. And that strong precursor-specific connection just wasn't really there in the KRAS wild type cases.
So if you have a KRAS mutation, which is already a big step towards cancer, this inflammatory signal isn't just a push. It's like a turbocharger.
It is a turbocharger, and it's acting right at the very beginning. The KRAS mutant precursor spots had way higher levels of key inflammatory pathways, especially NF-kappa B signaling.
So the oncogene and the niche are working together. KRAS sensitizes the cell, and the inflammation provides the go signal.
That's the synergy. It's a perfect storm.
Okay, so we've identified the cell, the niche, the specific pathway, and the genetic link, all happening in the earliest phase. This feels like such an obvious therapeutic target.
It does, and the functional validation in the mouse models really backs this up.
Tell me about that.
So they used a mouse model that developed KRAS mutant tumors. And crucially, the KACs in these mice also showed the highest expression of the IL-1R1 receptor, the same lock we see in humans. The biology was conserved.
And when they actually tested the signal?
They did these beautiful experiments with organoids, which are like mini precursor lesions grown in a dish.
Right.
When they added IL-1 beta to these organoids or co-cultured them with the inflammatory macrophages, they saw a huge increase in the number and size of the organoids.
Direct proof that the signal makes them grow.
Direct functional proof. And even more if they genetically deleted the receptor IL-1R1 in the KRAS mutant mice tumor development just plummeted.
Evidence is really mounting. So let's talk about the actual therapy. The timing seems critical.
It's everything. So they tested an anti-IL-1 beta antibody against an anti-PD-1 antibody, which is a standard immunotherapy.
And they tried it at 2 different times.
Yes. A prevention window right after the mice were exposed to a carcinogen, and an interception window starting a few months later when precursors were already established.
And the results.
The results were clear. The anti-IL-1 beta monotherapy was significantly more effective than anti-PD-1 monotherapy at reducing tumor volume.
So attacking that early inflammatory foundation is more important than attacking the later immune escape mechanism.
In the early stages, yes. But the combination was the real winner.
What did that do?
The combination of anti-IL-1 beta and anti-PD-1 gave the greatest tumor suppression, especially in that interception phase. It did 2 things. It shut down the pro-tumor inflammation, and it boosted the number of active, cancer-killing T cells.
So you're turning off the growth signal and turning on the kill signal at the same time.
Exactly. It's a 1-2 punch.
But this all comes back to that narrow time-sensitive window.
It does. And this might be the most critical finding for any future clinical use. They did a control experiment where they tried giving anti-IL-1 beta to mice with fully established invasive LUAD tumors. It did nothing. It completely failed to reduce tumor growth.
It's useless if you wait too long.
Utterly. It just confirms that you have to target this pathway during those precancerous stages when the IL-1B, IL-1R1 niche is actively driving the whole process.
So to just quickly synthesize this whole roadmap for you, the listener, LUAD starts with these specific alveolar progenitors, the KACs or RPII. They're unstable.
But that instability only turns into cancer when those cells are activated by a unique, stage-specific inflammatory signal from their neighbors, a signal driven by the IL-1B, IL-1R1 axis.
And that defines a very discrete, very vulnerable therapeutic moment. The early cancer is literally dependent on that niche to survive.
It gives us a tangible target for interception long before a real tumor ever forms.
You know, this research actually explains a major medical mystery from a few years ago. Do you remember the CANTOS clinical trial?
Of course. The cardiovascular trial using canakinumab, an IL-1 beta antibody.
Right. And they found, completely by accident, that it reduced the risk of people getting lung cancer in the first place. But then
later trials that tried to add that same antibody to treatment for advanced LUAD failed spectacularly. It provided 0 benefit.
And that confused everyone. Why would it prevent cancer but not treat it?
The spatial roadmap explains it completely. It works when the IL-1B, IL-1R1 niche is active during the precursor, the incident phase of the cancer. Once LUA is advanced, the cancer has moved on, and blocking IL-1 beta is like closing the stable door after the horse has bolted.
It really forces us to rethink everything about high-risk screening and prevention. The question is no longer just, can we treat advanced lung cancer?
No, the question now is different.
The question is, can we screen high-risk people, especially those we know have KRAS mutations, for the specific inflammatory signature, and can we extinguish that fire before a single tumor even forms? That feels like the next great frontier.